The review highlights the active components of some of the plants and their potential anti-cancer properties. It also delves into the role of apoptosis, the process of cell death, in cancer treatment. Additionally, the document explores the significance of signal transduction pathways and growth factors in cancer cells. The review emphasizes the importance of understanding the genetic and cellular mechanisms of cancer for developing effective therapeutic interventions. The key causes of cancer are multifactorial and can include genetic factors, environmental exposures such as tobacco smoke, radiation, and certain chemicals, lifestyle choices like diet, physical activity and alcohol consumption, and infections. The role of plants as anti-cancerous agents lies in their natural compounds, such as phytochemicals and bioactive molecules, which have been found to exhibit anti-cancer properties. These compounds can interfere with various stages of cancer development, including inhibiting the growth of cancer cells, inducing apoptosis, and preventing the formation of new blood vessels that tumors need to grow. Additionally, some plant compounds have been shown to have anti-inflammatory and antioxidant effects, which can contribute to their potential anti-cancer properties. Therefore, the use of plant-based therapies in cancer treatment is an area of active research and holds promise for the development of novel cancer treatments. Oncogenes and tumor suppressor genes play crucial roles in the development of cancer. Oncogenes are genes that, when mutated or overexpressed, can promote the growth and proliferation of cancer cells. These genes can encode proteins involved in cell growth, survival, and division. When oncogenes are activated, they can lead to uncontrolled cell growth and contribute to the development of cancer. On the other hand, tumor suppressor genes act as regulators of cell division and growth. They are responsible for repairing DNA damage, controlling cell cycle progression, and promoting apoptosis in damaged cells. When tumor suppressor genes are mutated or inactivated, they lose their ability to control cell growth and division, allowing abnormal cells to proliferate unchecked, which can lead to the development of cancer. In summary, oncogenes promote the growth and division of cancer cells, while tumor suppressor genes act to inhibit the development of cancer by regulating cell growth and promoting cell death. Mutations or alterations in these genes can disrupt the balance of normal cellular processes, contributing to the development and progression of cancer. Apoptosis, or programmed cell death, involves several pathways that contribute to cancer treatment. The two main pathways involved in apoptosis are the intrinsic or mitochondrial pathway and the extrinsic or death receptor pathway. The intrinsic pathway is initiated by internal cellular stress signals such as DNA damage or lack of growth factors. It involves the release of cytochrome C from the mitochondria, which activates caspases, leading to cell death. In cancer treatment, promoting the intrinsic pathway can induce apoptosis in cancer cells, helping to eliminate them. The extrinsic pathway, on the other hand, is activated by external signals, such as the binding of death ligands to death receptors on the cell surface. This triggers the activation of caspases and ultimately leads to cell death. Targeting the extrinsic pathway can be a strategy for cancer treatment by inducing apoptosis in cancer cells. Both pathways converge on a common machinery of cell destruction involving caspases that cleave proteins and lead to the dismantling and removal of doomed cells. Understanding these pathways is crucial for developing cancer treatments that can selectively induce apoptosis in cancer cells while sparing normal cells. Targeted therapies that modulate apoptosis pathways are an active area of research in cancer treatment, aiming to develop more effective and less toxic treatments for various types of cancer. The paper review on cancer and anti-cancerous properties of some medicinal plants discusses various plants, their active components, and therapeutic applications. Here are some examples. 1. Agave americana, Agavaceae. Parts used: leaf. Active components: steroidal saponin, alkaloid, coumarin, isoflavonoid, hecaginin, vitamins A, B, C. Therapeutic applications: potential anti-cancer properties. 2. Polygonum cuspidatum, Polygonaceae. Parts used: whole plant. Active components: saponin, flavonoid, vitamin A. Therapeutic applications: anti-cancer properties. 3. Polygonatum multiflorum, Liliaceae. Parts used, whole plant, active components, saponin, flavonoid, vitamin A. Therapeutic applications, potential anti-cancer properties. 4. Potential kinensis, rolsaceae. Parts used, whole plant, active components, gallic acid, tannin, therapeutic applications, anti-cancer properties. 5. Pyrus malus, rosaceae. Parts used, bark and fruit, active components, quercetin, catechin, flavonoid, turmeric and gallic acids, fluorizin, procyanidin, therapeutic applications, potential anti-cancer properties. These are just a few examples of the plants, their active components and potential therapeutic applications discussed in the paper. Several active components from the discussed plants play a pivotal role in the elimination of cancer. Some of these active components include 1. Flavonoids. Flavonoids found in various plants, such as quercetin and rutin, have been shown to induce apoptosis in cancer cells. They have been identified for their potential anti-cancer efficacy and ability to inhibit carcinogenesis. 2. Curcumin. Curcumin, found in turmeric, Curcuma longa, has been extensively studied for its potential anti-cancer properties. It has been shown to inhibit the generation of reactive oxygen species and interfere with the efficacy of chemotherapeutic drugs that induce apoptosis. 3. Saponins. Saponins, such as those found in Agave americana and Polygonum cuspidatum, have been studied for their potential anti-cancer properties. 4. Tannins. Tannins present in Potentilla chinensis have been associated with potential anti-cancer effects. 5. Quercetin, catechin and other flavonoids. These compounds found in Pyrus malus have been studied for their potential anti-cancer properties. These active components have been identified for their ability to induce apoptosis in cancer cells, inhibit carcinogenesis, and interfere with cancer cell growth. The active components found in the discussed plants have been studied for their mechanisms in eliminating and suppressing tumor cell growth. Here are some ways in which these active components exert their effects. 1. Induction of apoptosis. Many of these active components, such as flavonoids, curcumin, and saponins, have been shown to induce apoptosis in cancer cells. Apoptosis is a process of programmed cell death, and the induction of apoptosis in cancer cells can lead to the elimination of these cells. 2. Inhibition of carcinogenesis. Some active components have been associated with inhibiting the process of carcinogenesis, which is the formation of cancer. By interfering with the initiation and progression of cancer, these components contribute to the suppression of tumor cell growth. 3. Interference with reactive oxygen species, ROS, generation. Certain active components, like curcumin, have been shown to interfere with the generation of reactive oxygen species. Since some chemotherapeutic drugs induce apoptosis through the generation of ROS, the ability of these components to interfere with this process can impact tumor cell growth. 4. Regulation of signaling pathways. Active components may also regulate various signaling pathways involved in cell proliferation and survival. By modulating these pathways, they can influence the growth and survival of tumor cells. 5. Antioxidant and anti-inflammatory effects. Some active components exhibit antioxidant and anti-inflammatory properties, which can contribute to their potential anti-cancer effects by reducing oxidative stress and inflammation associated with cancer development. These mechanisms collectively contribute to the potential of these active components in eliminating and suppressing tumor cell growth, making them promising candidates for further exploration in cancer treatment and therapy. The findings presented in the paper have several limitations that are important to consider. 1. Limited clinical evidence. Many of the findings are based on preclinical studies, and there is a lack of robust clinical evidence to support the efficacy of these active components in human cancer treatment. More clinical trials are needed to validate their effectiveness and safety in human subjects. 2. Complexity of cancer. Cancer is a complex disease with various subtypes and genetic mutations. The specific effects of these active components may vary depending on the type and stage of cancer, and more research is needed to understand their precise mechanisms of action in different cancer contexts. 3. Bioavailability and pharmacokinetics. The bioavailability and pharmacokinetics of these active components are crucial factors that determine their effectiveness in vivo. Understanding how these components are absorbed, distributed, metabolized, and excreted in the body is essential for assessing their potential as anti-cancer agents. 4. Potential side effects. While these active components show promise in cancer treatment, their potential side effects and interactions with other medications need to be thoroughly investigated. Safety profiles and potential toxicities should be carefully evaluated. 5. Standardization of extracts. The standardization of plant extracts is essential to ensure consistent and reliable concentrations of active components. Variability in the composition of plant extracts can impact their efficacy and safety. 6. Regulatory approval. For these active components to be used as anti-cancer agents, they need to undergo rigorous regulatory approval processes. This involves demonstrating safety, efficacy, and quality control, which requires substantial research and investment. Addressing these limitations through further research, clinical trials, and regulatory processes is essential to advance the potential of these active components as anti-cancer agents and to translate preclinical findings into effective cancer treatments for human patients.